Okay, so we're going to be looking at some biochemistry viva questions, and we're going to specifically be looking at carbohydrate metabolism. So the first question you can be asked is the classification of carbohydrates. So carbohydrates can be classified as simple or complex. Under simple, you're going to say there is monosaccharides and disaccharides, and you can give examples. For example, in monosaccharides we have glucose, fructose and galactose, and in disaccharides we have sucrose, maltose and lactose. We also have the complex carbohydrates, for example polysaccharides, which are polysaccharides, and examples of polysaccharides are starch, glycogen, which is found in the liver and in the muscles, and fiber, which can also be classified as soluble or insoluble. So that will serve as our question number 1. Question number 2 that you can be asked is the digestion and absorption of carbohydrates. So for the digestion, we have to remember the enzymes, and here you can be asked of the difference between amylose and amylopectin enzymes. So amylose, its structure, we can differentiate these two enzymes in terms of structure, linkage, solubility and color with iodine. So the first one, structure. For amylose, it has a linear structure. Amylopectin is branched. And then for linkage, amylose has alpha 1 for linkage, while amylopectin has alpha 1 for linkage and alpha 1 6 linkage. Solubility, amylose is soluble in water. Amylopectin is sparingly soluble in water. For the color with iodine, we have amylose is blue with iodine, turns iodine blue, and amylopectin turns iodine violet. The other question under digestion and absorption is the process now. So the process of digestion of carbohydrates. In the mouth, we have starch digestion beginning, and the enzyme involved is salivary amylase. In the stomach, we have salivary amylase being inactivated because of HCl. The pH is low, so salivary amylase cannot operate. The next one will be in the small intestines, where everything is going to be completed now. We have starch being converted to maltose, and the enzyme used is pancreatic amylase. So we have converted a polysaccharide to a disaccharide, and the enzyme we're using is pancreatic amylase. And then this is a place where digestion is going to be completed, and the enzymes involved here are the disaccharidases, for example sucrase, maltase and lactase. Sucrase converts sucrose to glucose and fructose, and remember these are hydrolysis reactions, therefore we're going to be splitting the water molecule. So sucrose to glucose and fructose, maltose to two glucose molecules, and lactose to glucose and galactose. The still under question 2 chose digestion and absorption. Now we go to the absorption part C. Absorption of carbohydrates. Simple carbohydrates diffuse across the intestinal wall to other tissues. So the simple ones can easily diffuse across the wall of the intestines, and they can go into tissues like the liver, and there they're converted into fatty acids, amino acids, or glycogen for storage. Okay. Next question under carbohydrate metabolism will be glycolysis. And you know that glycolysis takes place in the cytosol of the cytoplasm. And then glycolysis is also known as EMP pathway, which in full would be embedded Meyerhoff-Parnas pathway. And the meaning of glycolysis is that it is a process of oxidation or breakdown of glucose to produce energy. Process of oxidation or breakdown of glucose to produce energy, which is ATP. So the first part was meaning. The second part will be the overall reaction or the energy derived. So glucose, we're going to the reaction involves glucose plus 2 ADP molecules plus 2 inorganic phosphate molecules plus 2 NADP, plus 2 NAD plus to give you 2 pyruvate molecules, 2 ATP molecules, 2 NADH molecules, and 2 H plus, 2 hydrogen ions in short. And then remember that 1 mole of glucose is going to give you 2 moles of pyruvate, and you're going to get 6 or 8 moles of ATP in the end. And then the other question under glycolysis will be the uses of NADH. Remember, in the reaction we're forming NADH. So what's the purpose of this NADH? What's its use? So the first use will be it's used in oxidative phosphorylation in the mitochondria, and we have the ETC, electron chain pathway, which in complex 1 we're going to use 2 NADH molecules, remember the ones we formed here in glycolysis, 2 NADH molecules plus 2 hydrogen ions plus oxygen to form 2 NAD plus molecules and water. So the first use is that it's used in oxidative phosphorylation in the mitochondria. The other one would be it's used in generation of NAD plus required for glycolysis. Remember also this complex 1 for the ETC is called NADH, ubiquinone oxidoreductase. Okay, so generation of NAD required for glycolysis. And this, where is this NAD plus used in glycolysis? Reaction 6 of glycolysis, where glutaralaldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate in a reversible reaction using the enzyme glutaralaldehyde 3-phosphate dehydrogenase. And here NAD plus, which we have just gotten from this, is going to be reduced to NADH and to NADH in short. Then the other question would be types of glycolysis. So we can say that there are 2 types of glycolysis: aerobic glycolysis and anaerobic glycolysis. So aerobic glycolysis is the glycolysis which takes place under aerobic conditions, and of course aerobic conditions involve oxygen, and the dominant product produced is pyruvate. So here glucose in the presence of oxygen is converted to pyruvate, and later this pyruvate is going to go to the TCA cycle. And then for anaerobic glycolysis, we have lack of oxygen, for example in strenuous exercise, and and the dominant product is lactate. So how do we get lactate? So glucose is going to be converted to pyruvate in absence. Okay, in this case we don't have oxygen, and then pyruvate is converted to lactate, and then so we want to focus more on this pyruvate to lactate reaction. So pyruvate is converted to lactate using the enzyme lactate dehydrogenase, and then lactate goes into circulation, and this process of pyruvate to lactate is going to reduce NADH and give us NAD plus. And you remember what's the use of NAD plus? Oh, sorry, we've not talked about the use of NAD plus, but this NAD plus is going to be useful, and we're going to see that very soon.